Chapter 10, the structure and function of DNA. So back in the 1930s, it was pretty clear that one type of biological molecule was the basis of inheritance. One big debate was whether it was protein or a type of nucleic acid. And one of the reasons proteins was thought to be a potential molecule of inheritance was because it's very abundant in cells and really diverse in terms of its structure. In the 1940s, more research showed that chromosomes consisted of two types of molecules: DNA, deoxyribonucleic acid, there's the D and the N and the A, and proteins. So it was becoming clear that possibly DNA was the molecule of inheritance, and that was really verified in the 1950s with further experimentation that DNA clearly was the molecule of inheritance. So in a way, that's not that long ago, in the 1950s. And this ushered in the field of molecular biology, study of heredity at the molecular level. And we talked about DNA a bit already. So the effort to figure out the structure of DNA also occurred in the 40s and then into the 50s. And scientists identified its atoms and how they were bonded to each other, covalently and hydrogen bonding. And what they actually needed to do was to figure out what the exact configuration of the models to form the three-dimensional arrangement of DNA that would allow it to do what it does. So we know that DNA is able to store genetic information, copy that genetic information, the replication part of DNA, and how DNA is able to pass on its genetic information from generation to generation, from cell to cell, or from organism to organism, right? Offspring. So it was a big challenge to determine what is the actual shape of the molecule DNA that allows it to do these really important functions. So DNA and RNA, they are both nucleic acids, and they are long polymers. If you remember, a polymer is a long molecule made up of smaller subunits called monomers. So DNA and RNA can be referred to as polynucleotides in the sense that they are long. Poly means many, long chains made up of the individual nucleotides, which are the monomers. And just a little diagram here showing you RNA and DNA next to each other. We'll talk about the monomers and the structure in more detail in a little bit. And then the polynucleotides, the long molecules of DNA and RNA, have any sequence of 4 particular nucleotides. So we're talking about DNA here. And the nucleotides, we're abbreviating them as A, C, T, and G. And those initials stand for adenine A, cytosine C, thymine T, and guanine G. So those are just the 4 different polynucleotides identified by these letters, and they're actually the letters of what are called the bases of the nucleotides that we'll get into. And you notice over here on the left for RNA, it also has 4 nucleotide bases. They are C, G, U, A, and U. The main difference between DNA and RNA in terms of these nucleotide bases is RNA has the U base uracil, excuse me, while DNA has the T base thymine. But again, we'll talk about that more in a bit. Okay. So the nucleotides are joined by covalent bonds in a repeating pattern of sugar to phosphate, to sugar to phosphate, to sugar to phosphate, and that forms what's called the sugar-phosphate backbone that's represented here by this gray ribbon-like structure, whether it's dark or light gray here. It's just color-coded. Okay? And if you look here, in this model from your textbook actually, if you consider just one of these ribbons, Here in blue and in yellow are the alternating sugar and phosphate, sugar and phosphate, sugar and phosphate, and that represents, or is represented by this strip, and that is the sugar phosphate backbone, as we have it labeled here, sugar phosphate backbone. Right? And that's a repetition of the previous slide. Okay, all I did is I showed a new figure here for you. This one's actually out of textbook. Here's the DNA molecule as represented as a double helix that we'll talk about. But if we're focusing just on one half of the helix, the ribbon-like structure represents the sugar phosphate backbone, and then coming out of it, branching out of it, like kind of like rungs on a ladder, if you will, are the bases of each nucleotide. So each nucleotide, as you see over here, has a phosphate, sugar, and a base. The phosphate group and the sugar group of each nucleotide is the same, identical. The only thing that's variable are the nitrogenous bases, right? The A, C, T, and Gs. Okay, so here's a little video showing you the two representative nucleic acids, DNA and RNA, just a really relatively short section of each one. And let's watch this video here.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group.
This numbering of the carbons in the nucleotides for DNA and for RNA, you don't need to worry about that. I will not ask you that at all. What I do want you to know is that the phosphate is covalently bonded to its particular sugar in RNA and DNA, and then that sugar is covalently bonded to the next phosphate and such in the sugar phosphate backbone. Those are covalently bonded components of the nucleotides, all right? And they are also covalently bonded to their nitrogenous bases.
DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide.
And again, he's talking about the covalent bonding, as I just mentioned, that I need you to know, but do not worry about the 5' and 3' ends or the 5' 3' carbons. Do not worry about that detail.
RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
All right. So notice in this representation here of the DNA double helix, because you have two strands that are bound to each other through the multiple hydrogen bonds, the little dots that you see here between the bases. And the bases, remember, they're one component of each nucleotide. So they don't show you the sugar and the phosphate groups. They're just represented by the blue ribbon here. What they are showing you in color code or color coded are the bases that stick out from the sugar phosphate backbone, and the little dotted lines indicate that the bases are covalent, that the bases are hydrogen bonded to each other. So the little dot, dot, dot represents the relatively weak hydrogen bonds. And then the base pairings are always complementary. T always binds with A always binding with T, or G binds with C, C binds with G. So they're complementary to each other because of their structures, the bases are. But notice that because they are complementary to each other, if you know the base sequence of one strand of DNA, you can easily determine the other base sequence because they are complementary to each other. And that's a key component of the structure of DNA that allows it to carry information and allows it to be replicated and allows it to take one of those copies and pass it off to the daughter cells or to the actual offspring for sexually reproduced organisms. All right, so the nitrogenous bases are arranged kind of like ribs on a backbone, if you consider it this way, or rungs off of a ladder. Here's just a single strand of DNA. And again, normally DNA is double-stranded. So kind of like half a ladder, or half of a ladder, with the sugar and phosphates making up the sugar and phosphate backbone, the sides of a ladder, and then the nucleotides are sticking out kind of like the rungs of a ladder. So each nucleotide has three components: the nitrogenous base, in this case it's thymine here, T, or A adenine, C cytosine, or who's missing, G for guanine, okay? And the sugar, deoxyribose for DNA, and then the phosphate group shown here in yellow. Okay, so that's one monomer, the DNA nucleotide with its three components. So the nucleotides in DNA differ in their nitrogenous bases, right? There are two types. Thymine and cytosine are single ring structure, like that video mentioned, and then adenine and guanine are larger double ring structures. This is only showing you the thymine, meaning a base with only a single ring structure. And then if you remember in terms of base pairing, excuse me, A always binds with T, and G always binds with C to form the double helix of the DNA molecule. I had a student tell me the way to remember this: good couples, GC, are always together, AT, if that helps. All right, so DNA and RNA have the same chemical structure, meaning they're made up of nucleotides, very similar chemical structure, except for DNA has the base thymine on one of its nucleotides. However, RNA has the base uracil on one of its nucleotides. So that's the main difference. Okay? Also structurally, DNA is usually double-stranded. RNA is